Grafting. In this method of reproduction, two plants of closely related varieties are joined together so that they will grow as one plant. Here the plant which is fixed to the soil is called a stalk, and the one which we are inserting is called a scion. So both stalk and scion, the cambium means, did you remember, the intercalary meristematic tissue. So that tissue will mix it and grows as one plant. Okay, here I am going to share one video. Next one is tissue culture. This method is called micropropagation also, because it is used in a miniature form. So many plants can be produced in a small test tubes. And what is the main feature behind it? Main principle behind this is every cell, plant cell, is having high dividing capacity, that is called totipotency. So based on that, this idea is developed. But what is the disadvantage? This cannot be done by the farmer himself in the farmland. This can be done only in the highly controlled conditions. So I am going to share a video of this.
Plant tissue culture. Cutting and grafting are the improved methods of natural vegetation propagation in plants. In 1902, G. Haberland noticed that the plant cells can be grown in synthetic media. The discovery by Haberland that the plant cells have the capacity to grow in a nutrient medium in presence of sufficient light made an impact in plant propagation and crop improvement. The ability of a plant cell to give rise to a whole plant is called totipotency. The cell and tissue culture techniques have taken the method of asexual reproduction in plants. In this method, instead of taking cuttings from a plant, a cell or a few cells or a tissue is taken from a plant and cultured in suitable containers in which nutrient medium is present. The portion of the plant that is taken as the desired plant is referred as explant. By this method, thousands of plants can be produced from a single desirable parent in one generation. These techniques require maintenance of sterile conditions in the medium. The explant needs to be sterilized before it is introduced into the medium. The medium is kept free from bacteria and fungi. Cells or tissues are grown in culture under a set-like condition. In the tissue culture, the explant often divides to give rise to an unorganized massive tissue called callus. The callus or the explant is such will undergo differentiation into roots, shoots, or embryo-like structures is called embroids. This differentiation is dependent on the concentration and combination of the plant's growth substances like auxins, cyanins, gibberellins, etc. In the medium, plants raised through tissue cultures are routinely used in agriculture, horticulture, and forestry. We'll use a diploid explant. We get a diploid plant like in any other vegetative propagation methods. Similarly, if we want to develop a haploid plant, it is possible to obtain it through tissue culture. Development of haploid plants through tissue culture was discovered by Indian scientists Shipra Guha and Satish Maheshwari.
Vegetative propagation advantages and disadvantages. So advantages are, so many advantages are there. What are they? It is quicker, easier and faster method of plant reproduction, and also it helps in raising the plants which are identical to the parent plant. So that's why these are called clones. Then it is the method to produce the plants without any viable seeds, like a banana. Banana do not have seeds. So such plants can be easily grown by this vegetative propagation. And what is the one more advantage? They bear fruits and flowers earlier than those produced from the seeds. So these are the advantages. Next, what are the disadvantages? As this method produces genetically similar plants, diseases of parent plant may affect the offspring. So that is the one biggest disadvantage. That's why we have to select the parent plant, a good healthy variety. Then the plants are nearest, as their plants are nearest to the mother plant, so there is no dispersal mechanism. Because of that, they compete for resources and a few of them may die. Next, third one is no variations. So no variations cannot be generated or created. That's why it may become fatal to the so many organisms. So these are the disadvantages. When I was giving introduction about how do organisms reproduce, that time itself I told that spore formation where I have to place it. So that's why I am taking this after the vegetative propagation. Spore formation is the example for fungi. Fungi will reproduce by spore formation. Next to pteridophytes like a fern. Fern also will reproduce by spore formation. Actually our bread mold, mucar, agaricus, these are all will produce by spores. So I will share one video. And what is the advantage of this spore formation? Spores are minute, light, and that's why they can easily fly in the air, and they also produce more in number. That's why easily disperse in the air, and they have a thick cell wall which is made up of chitin. Chitin, did you remember? The fungi cell wall is made up of chitin. So this chitinaceous cell wall will protect them till they get favorable conditions. So that's why whenever bread piece you keep it moist, or a little water droplets are there, and if you cover it, immediately the fungus will grow. So spore formation, example bread mold.